Hi everyone, my name is Andrew Ortiz. I am a fourth year neuroscience PhD student in the Department of Brain Health. Before I begin, I want to say thank you very much for taking the time for you to visit my poster. I really appreciate it. Now, today I'm going to be talking about a data set from a project that I've led for the past 3 years that involves investigating risk factors, as you can see in my title, such as diabetes, that significantly increase the likelihood of developing Alzheimer's disease. So for some background, very briefly, Alzheimer's disease is a brain disease that leads to progressive brain cell death, which eventually leads to learning and memory deficits and cognitive and behavioral impairments. And it is arguably one of the most devastating diseases that can affect an individual as well as their friends and family. So something that we see in Alzheimer's disease patients is a pathology called neurofibrillary tangles that are composed of accumulated hyperphosphorylated tau protein. Now, tau is a protein that holds the brain cells' communication structures together. And in Alzheimer's, when they become hyperphosphorylated, the protein starts to break off from these structures and then they start to clump up with each other or aggregate inside of the cell, causing them to form like this plaque that eventually destroys the brain cell and causes it to collapse and die. Now, aging is the largest risk factor for Alzheimer's disease, and diabetes is also a major risk factor for Alzheimer's disease. So, people with diabetes are up to 4 times more likely to develop the disease, and intriguingly, 80% of people who do have Alzheimer's disease have type 2 diabetes or glucose or sugar intolerant in the brain, meaning that they're not able to properly utilize sugar or energy correctly. Now, you're gonna hear me say the word hyperglycemia a lot. So hyperglycemia just means um, high sugar levels in the blood, and that is a primary characteristic of diabetes. Now, the mechanisms by how aging and diabetes confers increased risk for developing Alzheimer's disease remains unknown, and that is what my research aims to elucidate. Now, several studies have confirmed that hyperglycemia can initiate an immune response in the brain, and that can exacerbate Alzheimer's disease pathology, as I mentioned previously, like the tingles, and learning and memory deficits that are consistent with Alzheimer's disease. However, it is unknown if you look at underneath my introduction, that table, it is unknown whether hyperglycemia itself, like it's unknown whether the high sugar levels themselves is causing these deficits or if it's the neuroinflammation as a result of the high sugar levels that are causing these deficits. So this project that I did aims to answer that question. Again, whether it's the high sugar levels themselves that is increasing Alzheimer's disease pathology, or if it's the inflammation as a result of the hyperglycemia that's increasing the Alzheimer's disease pathology. Now, in order to investigate this, I had to utilize two different drugs. So the first drug is streptozotocin. I'm going to say STZ. So STZ is a drug that induces hyperglycemia by destroying pancreatic beta cells that produce insulin, so thus preventing glucose from exiting the blood. Secondly, another agent was used called fluoxetine or PZ. So PZ is a drug that lowers sugar levels by preventing the small intestine from absorbing the sugar and also the kidney from reabsorbing glucose. So basically, the extra sugar in the blood gets urinated out. So the idea is what we wanted to do is we wanted to induce a hyperglycemic state in one experiment for the entirety of the experiment. And in another set of group, we induce hyperglycemia, but we stop the hyperglycemia after a sustained incubation period, which hypothetically would stop the inflammation. And then we wanted to see if the Alzheimer's disease pathology continues to increase. If it does not, it would indicate that the neuroinflammation is driving the force that is increasing Alzheimer's disease pathology. So to answer this question, we utilized 24 young or 7-month-old mice against aged or 24 aged or 18-month-old mice, wild type mice. So there was 4 different treatment groups. There was a control group that received no actual drug, but instead just a vehicle. The second group was the STZ alone, so the hyperglycemic group. The third group was the PZ alone group, so again, the drug that lowers the glucose levels. And then lastly, the STZ plus PZ group was the group that was hyperglycemic for a sustained period of time, 9 weeks, and then they received a drug to lower the glucose levels. So if you look at my figure 1 and 2, this is a proof of concept. These are tracking blood glucose levels throughout the weeks um, across all four groups. Essentially, it's just proving out that both of the drugs are working. So the mice or the groups that received STZ did in fact have elevated glucose levels for both the young, which is in the left, figure 1, and the age, figure 2. And then if you look at the arrows, there's like a big arrow on week number 9 for both of the graphs. That is when we introduce the drug that lowers hyperglycemic to the respective groups. And as you can see, their glucose levels drop. The criteria for a hyperglycemic or diabetic state is 250 milligrams per deciliter. That's the criteria in the literature. So we see that STZ groups show significantly increases of blood glucose levels in both cohorts. And again, when we introduce the PZ, it lowers glucose levels. Again, for the STZ-PZ group, we are removing the hyperglycemic state to see if the inflammation in the brain continues to persist. Now, if you look at figure 3 and 4, we measure the amount of hyperphosphorylated tau, which is consistently seen in human Alzheimer's disease. We did this in the hippocampus, which is a brain region involved in learning and memory, and it is the first brain region affected by Alzheimer's disease. We did this in all the mice, and we basically saw that high sugar levels increased hyperphosphorylated tau, which is, again, the pathology for Alzheimer's disease in both young and aged mice. We also see that in figure 3 for the young mice, when we administered PZ to the hyperglycemic state group, we did in fact lower hyperphosphorylated tau levels. But this was not the case for the aged mice in figure 4. In fact, the hyperphosphorylated tau increased when we tried to decrease the hyperglycemic state. So this suggests that aging is leading to more susceptibility, or is more susceptible to fluctuation of glucose levels or sugar levels. So again, this is just preliminary results from my project. There are still several assays I need to conduct on various brain regions and peripheral tissues that are relevant to Alzheimer's disease, diabetes, and aging that still need to be done for this project, and I'm excited to share what we'll find in the future. Now, lastly, I wanted to say why is this research, or what are the real life implications of this research? So currently, there are 6.2 million people in the U.S. affected by Alzheimer's disease. Last year, the United States, we spent $305 billion to treat Alzheimer's disease, and by 2050, this number is projected to increase to $1.1 trillion. Secondly, is that there are 1/3 of our population, or 33%, have either diabetes. In the US population has diabetes or prediabetes, both of them risk factors for Alzheimer's disease. Lastly, by 2050, the US population is projected to increase to 25% to 400 million people. So with all that being said, given the increased trajectory of diabetes, Alzheimer's disease in population, and aging, it is important to understand the biological mechanisms by which diabetes and aging is conferring the increased risk for developing Alzheimer's disease, and that is what my project and research is focused on. Thank you very much for your time, and I hope you all have a good day.